Hello. Let's just get started. Thank you, everyone, for being here today. I'm Jose Avalos. I'm an assistant professor from Chemical and Biological Engineering and also here from the Andlinger Center for Energy and the Environment. Just a few reminders before we begin to please silence your cell phones. When the Q&A session starts, please wait till you get the microphone to ask your question. And then finally, and this is because we're recording this talk, so that way we'll be able to record your question. And then also, just a reminder, the next highlight seminar will be on April 18. Sally Benson giving a talk entitled Energy Systems Integration for Deep Decarbonization. So without further ado, I'd like to introduce our special speaker today, the highlight seminar given by Tim Donahue. So he is the Foundation Fetzer Bascom Professor of Bacteriology of the University of Wisconsin in Madison, where he's been faculty member for about 33 years. So Tim studies how microbes can be used to harness energy and carbon from biomass to make useful chemicals and fuels. He is the current secretary and past president of the American Society for Microbiology. And since 2007, Tim has served as a director of the Great Lakes Bioenergy Research Center, or GLBRC. This is one of 4 DOE energy centers to produce renewable fuels and chemicals from biomass. And he's also the interim director of the Wisconsin Energy Institute. So today he will be telling us about how the GLBRC is helping us get on the road to sustainable production of fuels and chemicals from biomass. So thank you very much, Tim, for being with us.
Thank you, Jose, and thank you to the Institute for inviting me here. It's been a while since I've been at Princeton, and it's always good to come back and connect with some old friends and make some new ones. So what I want to do today is lay out a vision for where we could be as a society for producing fuels and chemicals from the non-edible part of biomass, to replace the compounds that we currently get from petroleum or fossil fuels. And to do that, I want to begin with a few opening slides to frame the issue. These slides may be like me bringing coals to Newcastle, no pun intended, when we talk about renewables, but I want to put the challenge that we're trying to address in some context with these slides. So here's a first slide that I've stolen from a government agency that shows us how many BTUs of energy we've been using in the United States going back to its inception. And a couple of points, big picture points from this slide. The first is, not surprisingly, it's an ever-increasing number. The second point is that in the old days, biomass, the yellow line, was the predominant form of energy. And that's because we used to burn trees for heat and electricity, right? And then the fossil fuel industry kicked in, and now, as well as nuclear, and now in 2015, where this graph ends, we're at a point where this number is very big, and 81% of it comes from petroleum, gas, and coal. So people often say when I give a talk like this, well, that's 2015. Where are we today? We're doing all this other stuff in the renewable space. Where are we today? And so it's not on this graph, but here's another slide that I've stolen that shows in 2017, which is the latest data, they haven't published, to my knowledge, the 2018 data yet, we're still at 80%. That happens to be the lowest percentage in the last 100 years. So we're moving in a direction, but we're moving against a very strong headwind of a large number in fossil fuels. And if we come back to the biomass piece, biomass is, on this graph, shown as part of this 11% in renewables. And it's about half of that renewable. So it's about 5% of the total. So since the Civil War, we've gone from being almost 100% biomass to 5% biomass. And the biomass amount is only increased a little bit. It's everything else is increased a lot. And this government agency's projections are that by 2040, we will continue this trend of using less petroleum, gas, and coal. And their good news is we might be at 75 or 76%. So that's moving in the right direction. But from a technology standpoint, the point I want to make with all of this is if this is a race, this is a marathon, it's not a sprint. The amount of headspace that we need to change is huge. And as we change it, it's going to take a long time. As somebody who's lived through my life listening to music on records and 8-track tapes and cassettes and CDs, and has moved to this bloody thing that we all have in our pockets as a way of listening to music, this technology is going to change a lot slower than things like the music industry. But hopefully we can work together and get to a place where we can diversify the energy grid for a large variety of reasons. And you all are in the throes of this race here, and we're doing similar things at Wisconsin. And one of the things I would like to take home from this visit is a sense that we could work together moving forward. And by making some new friends and having me talk about what we're doing, hopefully we'll find ways to make new connections moving forward. So I am, as Jose said, I am the interim director of the Wisconsin Energy Institute, which is housed in this building right here. For those of you who know the campus, the football stadium's right over here. And our mission is to help the state and society move forward in energy. And like you all, we have broken down our activities into the two major pieces of the energy sector, and that's the transportation piece, which in the US is about 1/3 of the energy grid, and the stationary piece, which is the other 2/3. And we collaborate, again, like you all, with experts on campus on policy, economic, and societal issues and instruments that will help us move forward in energy as we create technology. But we're a little different than you all, from the sense that I have. Of the $35 or so million a year research budget in this Energy Institute, most of it is in the transportation sector. And most of it is in the bio side. If I look at energy institutes around the US, and even as the government funds energy, energy has been, and maybe will be, mostly a physical science enterprise, going all the way back to Edison and light bulbs and what he needed to power light bulbs, okay? So Wisconsin's a little different because we have a very large footprint in the bio field, in the transportation sector. And that's my segue to tell you a story about what we've been doing in this thing Jose talked about called Great Lakes Bioenergy. And because that's what I'm going to devote most of my time to today. So this is my outline of my lecture. I want to tell you who Great Lakes Bioenergy is. I want to tell you some things that have come out of our activities since 2007 when we were founded. And then I'm going to end with a slide or two on looking forward. And hopefully that will provide touch points for us to think about working together in this space. So who is Great Lakes Bioenergy? Great Lakes Bioenergy, as Jose said, is one of 4 bioenergy research centers established by the DOE Office of Science in 2007. We are now in our 12th year. This slide illustrates that this program has had bipartisan support from Congress through 3 administrations. It started with Bush 43, was continued by the Obama administration, and was just renewed for another 5 years with the addition of a 4th center by the Trump administration. So the Trump administration and Congress actually doubled down on their investment in this program last year. Each of these centers gets $25 million a year from the DOE Office of Science. So that means the investment by the federal government went from $75 million per year in the first 10 years to 100, about a 30% increase in taxpayer dollars. We all work towards the same goal, which is to generate basic science knowledge. We're an Office of Science funded center to convert the lignocellulosic or non-edible part of plant material into alternative sources of fossil-derived fuels and chemicals. So let me now put that sentence into some context. So the first is, why lignocellulosic biomass? So the federal government, namely DOE and USDA, estimate that the United States produces over 1 billion tons of lignocellulosic biomass each year on agricultural, forestry, and other acreage. You can think about that biomass in two major bins: residues from crops, so corn stover. I grew up in New York City, and 12 years ago, I didn't know what the heck corn stover was. So for those of you on the East Coast that maybe grew up in a big city like me, that's the stalks and the cobs, okay? So that's a crop residue. And more, we're shifting our focus into so-called dedicated energy crops. Things like woody biomass, which represents about a quarter of that 1.2 billion tons. So think of all the forests there are on the land. Think of the fact that the U.S. Forest Service is mandated by Congress to thin the forest in the national parks, and all the forest fires we have every year, because that hasn't happened. We could do something with woody biomass, either on public lands or for the paper and forestry industry by converting it into fuels and chemicals. And more, as you read the newspaper, you might hear about energy crops like switchgrass, other native perennials, and I'll explain why perennials are important, or sorghum as an annual crop that can be redeployed to make valuable products in it that Mother Nature hasn't had it made over millions of years of evolution. So there's a lot of biomass. Why is the conversion of that into fuels and chemicals something that we as a society are interested in? So DOE estimates from that tonnage, if we could convert half of it, not every gram, half of it, because some amount of it you might want to put back in the ground, we could replace 30% of the country's demand for fossil fuels and chemicals. So that's why I've taken these 3 barrels and put one of them in a green coat. That's the vision. Putting a third of what we get from petroleum in the game of lignocellulosic biomass. Remember that fossil-derived products are a multi-trillion-dollar per year industry in this country. So if we could convert a third of that product into biomass-driven fuels and chemicals in states like Wisconsin, the Midwest, and New Jersey, you could produce local value for a new industry in those states where the biomass is grown locally and made into high-value products locally. So in addition to all the environmental benefits, this is a jobs, local communities, advantage industries for people that grow and produce the biomass. So what is our mission? This is our mission statement, and it aligns very well with the DOE program goals: the sustainable production of specialty biofuels and bioproducts from lignocellulosic bioenergy crops. So let me explain what the asterisks mean. The first asterisk for specialty biofuels is not ethanol. We are charged or working to replace the petroleum part of your gasoline, not the ethanol, to replace the aviation fuel that I will fly home on, to replace the diesel in naval ships and in long-haul transportation vehicles. Bioproducts are substitutes for the fraction of the barrel that goes into chemicals currently, plus more if we think about the potential for synthetic biology to make chemicals that the petroleum industry cannot currently make out of a barrel of fuel. And lignocellulosic bioenergy crops is a mouthful word of saying we are not working on food crops anymore. We are not working on crop residues. We are working on engineering and productive and valuable energy crops that will be purposely grown for the production of energy and chemicals. So we're not competing with food production. So who are we? We get a lot of money from you all as taxpayers, $25 million a year. Here's a picture of many of us at our annual scientific meeting last May. There's about 400 of us now, scientists, engineers, students, and staff. Great Lakes Bioenergy is led by the University of Wisconsin. We have subcontracts at these 4 institutions. We have a special arrangement with Michigan State because probably a third of our research, $9 to $10 million a year, goes on in East Lansing, Michigan. But we have minor subcontracts at these other 3 places. We have programs in 3 major areas: the development of sustainable cropping systems, everywhere from where you grow crops, what you grow, what the energy and water and fertilizer inputs are into those crops, to how we modify crops so we have next generation energy crops when we can. We also have programs in efficient biomass conversion. How do you take lignocellulosic biomass, solubilize it, and create crops from as much of a ton of, create products from as much of a ton of biomass that shows up at the refinery as possible. And then we have a large set of activities that look at how these processes would need to be integrated in a conceptual biorefinery model. Everything from life cycle analysis to where would you place the refineries, what would they make, the techno-economic modeling of what the mix of fuels and products should be to be more profitable than a starch refinery and more like a petrochemical refinery, to molecular models of how the plants are going to deploy carbon to put into energetic materials, or how we can reprogram microbes to make the kinds of products that we want, to molecular simulations of the enzymes that we're going to need to optimize to be able to do that. So all of the knowledge in these 3 areas is acquired by sets of cross-cutting research teams. We are not a center that has a bunch of engineers working with a bunch of engineers and a bunch of microbiologists working with microbiologists and a bunch of agronomists working with a bunch of agronomists. We fund people to work as teams across those disciplines to hopefully catalyze creativity that comes from cross-domain activities that wouldn't happen if we worked in academic silos. And the other point on this slide is that everything that we do is driven by the economic, environmental, and technological sustainability in each of these areas. What are the most atom economical pathways that we can use? What are the best products to make locally or as a nation? And what are the best practices to maintain the sanctity of the environment where these crops are going to be grown so that we don't have another Dust Bowl 5 or 10 years from now? And we can do all of that with this amount of funding because it allows us to bring these teams of people together. So what I'm now going to do is spend most of my time talking about what we've done to set the foundation. And so I'm going to cut that into 2 major parts. The first is, what have we done that we think impacts the field? And then I'm going to give you some examples of what we've done at a high level to lay the groundwork for some of the challenges I just put out there. So first, If you were the Department of Energy and you came in and wanted to know what did we do with $267 million in the first 10 years before we were funded for another 5, you might have seen this slide. So this is our impact over the first, this is actually now 11 years because it includes data up until the 1st of 2019. 1,200 publications, which if you do the math is over 2 per week. Over 70,000 citations. This is great. We're academics, we produce knowledge, but DOE is a mission-oriented agency, and they want that knowledge to get out to the marketplace. Almost 200 patent applications on new types of crops, deconstruction methods, chemical and microbial catalysts, which turn out to be a little over 1 per month, almost 2. Over 110 licenses and options worldwide, which is 10 per year, and 5 startup companies. So this is the kind of intellectual ferment that we can produce with this kind of money. And if you look at these metrics and compare them to what happens per $1 million of grant funds for other agencies, we have outcompeted a lot of those other activities. And I think it's because of the importance of the problem and the openness of the field. And I also think there's been a lot of benefits to putting teams of people together the way we did in this program. This is my NASCAR slide. This is a slide that the people in Congress like to see because they say, well, who are you working with? So instead of giving them a list and telling them who they all are, we just show them the icons of the number and types of industries that we're actively working with in the center. And this represents everything from the combustion field, the engine manufacturers want to know what we're producing, to the biotech arena, who's going to make the fuels and chemicals, to the agronomic space, they want to know what kind of crops we're thinking about growing. And we work with them in a variety of ways. Some of these companies actually give us proprietary materials to evaluate in the center in the types of systems that we're rolling out. Some of them license technologies that we bring to our technology transfer agents. Some of them see what we build as a widget, and they say, well, could you evaluate that for us? And I know it's not part of your DOE mandate, but we'll give you some money to evaluate it under sponsored research. And some of them, there's about 12 people on this slide that serve on a scientific advisory board that we bring in every year to kick the tires, find out what we're doing, and give us advice on whether our programs are industry relevant. Let me make sure I put that in the right context. We are a taxpayer-funded center. We do what DOE tells us to do. We don't work for industry, but we like to know that the things that we're doing are addressing issues that industry thinks are at the forefront of the field in a way that they would not be able to address within industry themselves, because we are, after all, the crazy George Jetson academics that can take high-risk chances that you wouldn't take in industry, because you have a 2 to 3 to 5 year window at best to take a concept to product. And I'm going to show you some examples, I hope, of where that's been successful. So now let me spend a few minutes going through some high-level examples of concepts that have come out of what we've done in the past 11 years that are driving where we're going in the future. So the first set of examples I want to show you deal with defining what are sustainable energy crops. What should we grow over here? And how should we think about growing it? So the goal here was to identify and develop high-yielding, productive, and valuable cropping systems that would be feedstocks for industry to convert into fuels and chemicals. So if you think about that in the United States, you'll see there's a large number of cropping systems that you could evaluate. Anything from corn, which is low diversity, that's 1 species per acre, that has a high energy input. and chemical input because it needs fertilizer and it needs pesticide. At the other end of the spectrum are high diversity things like native or restored prairie that have a lot of different ecosystems on the land but have very little input for managing it because they're native. They don't get watered except by rainfall. People don't put fertilizer or pesticide on them. So what do we think are the best crops for us to deploy as energy crops? We think obviously they need to be productive. You need a lot of tons per acre. We think perennial crops are better because perennial crops, as a New York City boy, those are the weeds. They come back every year. You don't have to nurture them. They always come back. And the reason they come back is because they store a lot of carbon in the ground, in their roots. And they get big. So if you grow perennials, you can get a lot of biomass, and you can also sequester a lot of carbon in the root systems. We think polycultures are going to be a big player here. So that includes things like native prairie, native grasses that tend to grow not as a monoculture, but with their friends and their neighbors, and they figure out how to help each other create productive environments. And we think placement is the other critical issue that we need to consider as we think about the types of biomass crops. We do not want to replace acreage used and needed for food for fuels and chemicals, or else society will be in a different place, okay? So these are our 4 Ps: productivity, perennial, polyculture, and placement. And that's going to drive us in a few minutes to focus on energy crops that are grown on non-food, or the agronomic word is marginal land. So let me give you an example of one of the things that we've done taking advantage of the programs that we have to increase crop productivity. So the one example I'm going to give you is with switchgrass. And here's a picture of 2 types of switchgrass. The switchgrass you would grow in Wisconsin, which is lower yielding, and it's lower yield because the days get short early and it flowers, and then once it flowers, it stops growing. And you compare that to the switchgrass you would grow in the south, in the lowlands, which is higher yielding, it flowers late because the days are longer. That decision of flowering, which impacts yield, is driven by a molecular switch in the switchgrass that's been embedded in its circuitry over millions of years of evolution. And we wanted to flip that switch. Here's why we wanted to flip it. We wanted to flip it because our techno-economic analysis of a farmer being able to make money growing switchgrass predicted they needed to produce 10 tons per acre, so out here. And people for years had been looking for cultivars that they could move from Nebraska up to Wisconsin and see who survived, and they got wimpy and they froze in the winter and they never did well. So right after we were funded, we took advantage of working with the Joint Genome Institute and sequenced several lines of switchgrass, high-yielding and low-yielding ones, and began to look at the genetic differences and set up a genome-based evolution and breeding program for higher yield. And you can't really look for tall switchgrass. You look for ones that flower late. That's the switch that controls how much yield you get. And by doing that, you can now see after we did that, this is the increase in yield that we got once we set up this program. This is what we would have gotten if we kept doing the traditional breeding of crossing this line with that line as a way of getting improvements. And you can see that we had a dramatic rise in the rate of yield improvement once we did that. And we projected, even if we get no continued yield improvement at this rate, we could reach this 10 ton per acre target 25 years sooner than we would have if we just kept on this path. Now, my hope is we'll continue at a rate somewhere maybe as good at this, but definitely better than that, and we'll get to that point even faster. So we're learning a lot here on the plant physiology of what shuts them down when the days get shorter. That will be important for switchgrass, but we think we'll be able to take those lessons and transfer it to other crops down the road. Where would we like to deploy this knowledge? So here's an analysis, some of which is old and we've published, and some of which we're just starting, that talks about the potential for us to grow crops on so-called marginal lands. So to illustrate this concept, here's a map of the north-central region of the US, and it's a heat color-coded map with the dark green counties being more productive than the light ones in terms of tons of biomass per acre. So you can see that there's a lot of productive land in these dark green counties that is not used to grow food. Food is grown in those light yellow or light green counties. We estimate in these dark green areas that there's sufficient biomass for 24 million gallon per year biorefinery. 24 million gallon per year is kind of like the metric in the DOE models for what you need to build a biorefinery for to make it profitable. Each one of those biorefineries, if placed in the center of those circles, would be no more than 50 kilometers away from where it needs biomass. So you wouldn't be shipping biomass from North Dakota to Louisiana. You'd be growing it in North Dakota, shipping it 50 miles away, creating fuels and chemicals from that land, from that crop. So this is jobs. This is economic opportunities for communities. And if I was a chemical company and knew I was going to get chemicals from one of those refineries, I might want to build my chemical processing plant right nearby where they're making the chemicals that we're going to make. We also, thanks to the agronomy input in the center, know that the proper management of these lands can provide greenhouse gas, so again, carbon under the ground, and other ecosystem benefits. We do not want to clear these lands totally and ruin nesting sites for birds and animals, ruin the ability of biomass producers to hunt and fish and enjoy this land in the way that they normally do. So it needs to be managed, and we're developing an idea of how much should we take off per year at different sites, what would that allow a producer to do and the community to do to still enjoy that land while they make some money. And we know about the benefits of making fuels and chemicals from this ton of biomass. Just like the petroleum refinery industry makes half of their profit from making fuels out of 70% of that barrel and half of their product of making chemicals out of 15% of that barrel. So that's a totally different industry than an ethanol-only one-product industry. This other map here depicts what we're just beginning to do to evaluate the potential for this other acreage to host these types of lignocellulosic biorefineries as well, using dedicated energy crops. So if you were to overlay this map on this one, you will see all these green, red, and blue dots are over the light yellow circles, counties. So we have begun to take in satellite data from 8 years, over 8 million acres on this map, and overlay that with crop productivity data and weather data of how much water comes into these areas, and look at productivity in 3 ways. There's a lot of green on this plot, on this graph. Green is high productive farmland that's used for food. That's off the table for us to use for fuels. Red and blue are low productivity or non-productive land for food that we think we can turn into producing fuels and chemicals for society with very little energy input and very little chemical input. And if we did that, we would not reduce the food production capacity of that acreage very much, because they don't produce a lot of food right now. That's why many of those counties are very light colored. Okay, so let me move on, and I'm going to speed up a little bit and talk about what we've been doing to improve biomass deconstruction, because a ton of biomass shows up at the gate. We want to develop ways to take it apart into its various components so that we can ultimately convert it into fuels and chemicals. So we need low-cost, atom economical. Atom economical means we get access to as much of the carbon as possible, biomass deconstruction methods. Deconstruction is a major cost for any future industry. Probably half of the cost in making a gallon of fuel or a gram of chemicals is in biomass deconstruction. So changes here will have a large impact. So here's one of our breakthroughs, and it's based on the development of technology to make a compound known as gamma-valirolactone, which is this low molecular weight lactone here. GVL is on the top 100 list of DOE industry-relevant chemicals because it's a valuable commodity. It's a precursor for making various furans. So Jim Domesic in chemical and biological engineering figured out how to make this out of biomass. And we said, that's great. We now have a company started that's making GVL so that we don't have to import as much THF from Asia. Jim's postdoc said, given what I know about GVL, this might be a really good solvent to actually dissolve biomass. And he showed that that was right, that it dissolves biomass, and it actually gives us the two major components of biomass carbon, lignin and polysaccharides, in separate streams that we can process. And it's biomass agnostic. There are certain deconstruction technologies that work with grasses, but don't work with trees, and work with trees and don't work with grasses, because not all plant cells are created equal. Their cell walls are different. But GVL works with everything we've thrown at it, which is what you would want if you're a refinery, because then you can buy whatever biomass is cheap within that radius. You don't have a whole refinery based on grasses, and the grasses didn't grow this year, so I have to retrofit the whole refinery to deal with a different feedstock. And we've worked out techniques where we can recover over 98% of the GVL and reuse it again to solubilize more biomass. This works at nice little academic scale levels. We're now very excited about scaling it up and seeing if there's a way that we can make both of these streams in a way where they can then be used by downstream processes to make fuels and chemicals. But we're really excited about this moving forward. The other thing I'll talk about is some of the work in the discovery of enzymes that would degrade the sugar polymers, the polysaccharides. Again, this is a major cost in deconstruction. And right now, if you were running a refinery, there's two companies that supply you with enzymes that degrade the cellulose, mannose, and xylose cell walls of plants. And it's a magic little elixir that they won't tell you is what's in it. There's probably 15 or 20 enzymes in it that have each been optimized. And when we started and we told people we were going to go look for new enzymes, they said, you're crazy. Because industry has sunk tens of millions of dollars into this, and there's not going to be any new enzymes out there. Well, we discovered that there were bacteria that live on the nose of wood-boring beetles that destroy wood trees. And those bacteria secrete enzymes that have the ability to hydrolyze cellulose, mannose, and xylan, all three of the polymers, in one polypeptide chain. We've now discovered this is a whole new class of enzyme. We've done biophysical and kinetic analysis as well as structural analysis. Here's the structures of these. These gray areas are the active sites. They have evolved to be able to accept all three polymers. They're different than ones that only work on C or M or X substrates. So we're very excited about using these as a way to get simpler. Simpler means lower cost for industry, enzyme cocktails that you could use for biomass deconstruction. The last story I'll tell you about quickly is our efforts to convert all of the major biomass fractions into fuels and products. I've already mentioned there's two major fractions, the sugars that we are thinking about making in the one stream, and the lignin, which is in a second stream. The lignin stream is a polymer of aromatics. So I'm going to tell you what we've been doing to make products out of those. Our strategy is take the sugars, which is about 2/3 of the biomass, and make it in the fuels. Take the lignin and make it either in the chemicals or fuel additives. So let me give you a little progress report on where we are with that. So I'm not going to go through all we're doing to make the fuels. Jose is an expert on this, but we are focused on making things like isobutanol, which is a longer chain alcohol than ethanol, and it's an industry-accepted jet fuel. Terpenes and bio-oils, and here's a picture of an organism that's actually secreting lipids and lipid droplets into the media to overproduce them. When we make a fuel, or when we sit down and plan on making a fuel, we benefit from having an engine research group right across the street that will work with us to burn what we're planning to make at 10%, 20%, et cetera, either in an automotive or in a diesel engine mock-up at low scale. Sometimes, if the molecules we're making are commercially available, they just order it up from Sigma Aldrich before we even make it. Other times, they have to wait for us to make enough for them to test. The other thing we have the advantage of with this kind of activity in the center is the assembly of what we call a field-to-products pipeline, which allows us to grow biomass on our experimental stations at land-grant universities, pre-treat it in-house, hydrolyze it with enzymes if we need to. We make these things that we call hydrolysates. If you were making wine, this would be your grape juice. If you were making beer, this would be your mash. And then we ferment it and analyze it for output. At each one of these steps, we have a core facility that allows us to do chemical quantitative analysis of each of these fractions. So we know when we grow a crop how much sugar was in it, what those sugars are. When we process it, what recovery do we get by a standard procedure? When we run a fermentation, how does it work? We know when we make a hydrolysate and it does something bad to the fermentation microbe, we have data on its chemical composition for 100 molecules that we look at. We can then go to the chemical shelf and make a synthetic hydrolysate and ask if it behaves exactly like the real McCoy that we made from plants. If it does, it tells us something. And we can then make hydrolysates with 90 and see if we identify either something that's positive or an inhibitor. If it doesn't, it means we've missed something in the hydrolysate. We better go figure out what's in those other mass spec peaks that we haven't analyzed yet. So let me tell you some of the lessons that we've learned from doing that. So here's, I think, a really interesting and very useful example moving forward. If you think about how an industry is going to run, they're going to take in biomass at the end of every summer. and they're going to process it. Well, the weather's not the same every year. Some year it's dry, some year it's warmer, some year it's cooler. So is there an effect of interannual environmental variation on biomass? So once we set this pipeline up using biomass from 1 year, we started to look at biomass that we grew at a variety of years. So I want you to focus on 2012, which was a dry year. I'm going to call it a drought year. And we did this with corn stover and with switchgrass. So here's what happens if you make those sugar hydrolysates from switchgrass grown in 2012. And I want to draw your attention right here. This is a yeast fermentation on an optimized strain that we built that makes ethanol in 2011 switchgrass and 2013 switchgrass, but it makes no ethanol in 2012. And we figured out that there were furans in the drought year switchgrass that inhibited fermentations in yeast. Here's the same material fermented by a bacterial strain that we're developing as a biofuel producer. The bacteria don't care. They grow just fine and they make fuel. So if you were a refinery and you had a yeast process and you were buying switchgrass, you would make no money in 2012 switchgrass. Well, we have now figured out what the chemical is. We have figured out its target of action, and we have retro-engineered this yeast strain so that it would work even with 2012 switchgrass. So, and we also don't see this with corn stover hydrolysates. So this is a plant-specific response that only works on one type of biofuel-producing microbe. So the long and short of it is we have discovered that these lignocellulosic hydrolysates are replete with a wide variety of things that we call fermentation inhibitors or toxins. And we've identified a large swath of those. Some of them only work on yeast, some of them only work on bacteria, some of them work on both. Here's an example of a gene expression analysis in several dozen strains with several dozen inhibitors, and you can see that there's a lot of genes that are affected in all of these organisms by these toxins. Some of them are only affected by some toxins and not the others. This has allowed us to bypass those blocks and build performance-optimized strains that will still make fuel in the presence of those toxins. This is not making material from glucose or from cornstarch. This is making material from very complex hydrolysates. So the last thing I want to just say is I want to tell you a couple of little stories about making products from lignin. So why do you care about lignin? Well, it is 30% of the biomass carbon. So if you make no products from 30% of what shows up at the loading dock, your economics are going to be very different than if you can make products from it. And when we started, the other story we were told by industry was, that's a great idea, but we've thought and worked on this for a long time. And what we found is that you can make anything from lignin except money. So you're wasting your time working on lignin. So I'm going to tell you that that was bad advice, because we have been able to show that we can deconstruct and produce valuable compounds from lignin. So this is a tough nut. I'm not going to pick on the industry people too much. But this is what lignin looks like. It's a polymer of aromatic compounds. And the reason some of these are green, blue, and red is it's made from different precursors. And it's one of the few polymers in biology that is not put together with enzymes. It's put together with radical chemistry. So what that means is every aromatic polymer is different than its neighboring aromatic polymer, because it all depends on what radicals were there when the substrates got to the place where it's put together. So it's very heterogeneous. So we're working with people in the chemistry department. We've been able to show that we can chemically depolymerize this into lower molecular weight materials that we can then feed to microbes we've isolated that have a voracious appetite for these types of aromatic compounds. So here's an example. This would be the feed that we would give to this alpha proteobacterium. It's a bunch of stripes in different colors. I'm not going to bother telling you what they are, but they're different types of aromatics. And if you fed it to a wild-type organism, it would eat it, and all you would get out of it is more biomass. But if you go in and understand the aromatic metabolism pathway in this organism, you can make mutants. If you figure out how to make mutants, which we did, and you can see in this mutant, you get out almost pure hydroxybenzoic acid, and in this mutant, you get out almost pure benzoic acid. These are chemical feedstocks for the BTX industry, benzene, xylene, toluene. So we now have microbes that can take the mixture of aromatics that come from lignin, funnel them, and with the right genetic engineering, get them to make valuable aromatic precursors that the chemical industry will use. Another sidelight story that came out of this was the identification of this compound, poacic acid, which turned out to be one of the most toxic compounds we found in these solubilized biomass samples. And this is another one that was a specific inhibitor of yeast growth. And this shows the LD50 curve. This is poacic acid. It's an aromatic dimer. It's a diferrylate. And it's got a very potent LD50. We, by testing what was required, what the site of action of this compound was, we identified that it worked to inhibit yeast cell wall synthesis, in particular beta-1,3-glucan synthesis, and that put off a little red flag on our workstation because the patent literature said inhibitors of that enzyme were very, very important antifungals. Whether it's a yeast infection or whether it's a fungus that inhibits a plant pathogen or whether it's other animal or human fungal disease. So we've actually shown that when you treat soybeans with a known fungal pathogen with poacic acid, it protects against that native fungal pathogen. So here's an example of what could be a very potent antimicrobial that's come out of looking for energy products and biochemicals. The last point I'll make is that lignin is still one of the hardest things for us to deal with in the center. And that's because it's composed in a simple way of different colored units that are held together by very strong covalent bonds. And either enzymatically or chemically, we need to treat this compound really rough to get it to fall apart. But one of our investigators said, what if we could go in and do something to modify the lignin biosynthetic pathway and make it easier to degrade? And he coined this concept of zip lignins, where you would go in and instead of having these really tough covalent bonds to cleave, you put in twist ties. They would be easy to open and the aromatics would fall apart. This was a radical concept. It meant we needed to find an enzyme that no one had ever seen before. Well, we found that enzyme. And it meant we needed to actually do the first experiment in plant synthetic biology, where we replaced a crop, where we replaced a pathway that normally makes this type of lignin with an enzyme under the right promoter in the right type of cell so that it would make the zip lignin. The long and short of it, it works. We now have lignin that comes apart easier, lowers processing cost, gives me easier to work with lignin so I can make chemicals out of it, and demonstrates that we can actually go in and change the chemical composition of the plant lignin in the cell wall without having deleterious effects on the growth of that plant and its survival in the field. because we have this in trees. We're ready to harvest them after 3 years in Wisconsin, and they grow just as well as their wild-type counterparts. So we're really excited about this because it means we have the ability to make other types of lignin that industry might want moving forward. So with that, I'm just about done. Hopefully I gave you an idea of the kinds of major issues that we're working on and why I think it's okay for me to say that we're on the road to making fuels and chemicals from biomass. We don't have a refinery that does all of these steps yet. We're a university. We're not going to build a refinery. We're putting on a smorgasbord table options for companies to think about. Your crop, your product, what your economics are. The hope is that by maximizing the production of valuable biomass on marginal lands, and by converting as much of that into fuels and chemicals, and by being able to predict with TEA and LCA how these things will work, will generate value for producers, which in the Midwest is farmers, new refineries, consumers will be able to get locally low-cost products, and communities that will host these and have people move there to work in these sites, and provide economic advantages and markets for local industries in the US and elsewhere. So with that, I'm going to shut up. I think I got it through on time, and see if you all have any questions for me. Thank you for your attention.
Impressed with your talk.
Thank you.
Beautiful talk, beautiful idea. The most important is beautiful. Beautiful idea. However, my one question is, so I understand that you propose to make fuel by a million times faster than petroleum. In other words, making in few days instead of few million years, something like this first. But so also you create hydrogen, free hydrogen, correct? Or you create hydrogen? Can you trap this hydrogen as a fuel?
We are not creating a lot of hydrogen in the fuel fermentations.
No, so you don't worry about this one.
We did have a hydrogen program 10 years ago, but we shut it down.
However, another thing it is, you mentioned that carbon, to some degree, you eliminate carbon, you're putting carbon to the soil. However, your fuel is based on carbon mostly.
Yep.
So if it's based on carbon, you put in car and you create greenhouse gases. How you address this one, how you convince, say, people who claim that hydrogen, man-made hydrogen, carbon is greenhouse for which change climate? So
a couple of points. When you combust the fuel, you're absolutely right, you will release CO2 in the atmosphere. What we currently do now is we release carbon that was sequestered millions of years ago into fossil fuels into the atmosphere. This carbon was sequestered last year. So you're recycling with a different residence time is point number one. Point number two, if this is driven by a significant activity with perennial crops, if I go in and I harvest a 6-foot stand of switchgrass, there's 9 feet of carbon under the ground in the roots. So there is a lot of carbon that's been sequestered to give me that crop. Unless we electrify everything that we use for transportation, including jets and naval vessels and trucks, we are always going to be putting carbon into the atmosphere.
You have to create electricity, so.
Well, you're going to create electricity in a carbonized world also, unless we go to nuclear or solar. Yeah.
There's another question in the second row.
Yes.
Professor Donahue, very, very nice presentation.
Thank you.
So much. My question is, every approach has some pros and cons.
Absolutely.
I think you have kind of convinced us about the great benefits of using biomass for extracting fuels and chemicals. What are the big disadvantages? For instance, after all this extraction, you probably are going to be left with a lot of waste products. How do you deal with that?
Okay. So that's a great point. Let me first say thank you for giving me a compliment. The other point is maybe I've sold you, but I think we have a lot of challenges to realize this vision that I laid out. It's still a lot of work ahead of us. The biggest is, what are the molecules and can we do it at scale? Can we take what we do in an academic lab and do it in a 25 or 50 million gallon per year biorefinery? Residues. So you're absolutely right, and I didn't go through it, A, because I didn't have time, and B, because it's a new activity. But for those of us who drink and enjoy a glass of wine or a glass of beer, we know that ethanol is not the only thing in that glass. There's flavors and leftover organic material. And the same is true when you do a fuel fermentation or you do a lignin conversion. You get a product, but there's other organic residues. What industry would do with that now is one of two things, both of which provide very low return on investment. They would take that residue and dry it down and feed it to animals for feed, or they would take that residue and they would put it in an anaerobic digester and make methane, get electricity, use the electricity to run the refinery, and although that's a sustainable model, the cost of electricity is really low, and the functionalities in that organic material is very high, and we're actually rolling out new activities to take the conversion residue and make it into high-value chemicals. From what we've done and already published on, if we could convert 20% of the residue from a fuel fermentation into the products we're already making, we could drop the price of fuel by 25%. So we are thinking about every gram of biomass. I dealt with the lignin and the sugars, but as a microbiologist, we know every time you do a fermentation, you get leftover material, and we're going after the residues also. And that's a good thing, because there's people in the state of Wisconsin and elsewhere who have a lot of residue from food processing and agriculture. And the same organisms that work on these residues, we think will work on making valuable products from dairy and other types of residues. So.
Thank you. So the workflow that you presented seemed to show first biomass pretreatment and then hydrolysis, feeding hydrolysates, and then fermentation. Are you working at all on either simultaneous saccharification and fermentation or consolidated bioprocessing? We are not. We have chosen this strategy. There's one of our brother or sister centers in Oak Ridge, Tennessee, is set up their whole focus on consolidated bioprocessing, which is feeding the biomass to an organism that secretes the enzymes to break the sugars apart and then do the fermentation. Again, to come back to his question, you may think I have a great strategy. I'm not convinced it's a winner. That's what basic science is all about. There might be a company that uses my strategy or our strategy, or uses the Oak Ridge strategy, or takes a hybrid of something that we did and something that one of the other centers did, and builds their pipeline. So the jury is still out on what's going to really be deployed anywhere, if anything.
So you have showed the slide before that currently the market doesn't really use biofuels. So, so do you look at the problem from the demand side? So does the product actually require so much biofuel that you're producing, or changing it to 30%?
Yeah, we look at demand as part of our, quote, techno-economic analysis. And that's an interesting exercise for us, because I can evaluate the utility of making a compound. If it's currently already bought by industry and used. But I can, and so I can, then they can say it's worth so many dollars per ton and that we need so many tons per year, right? And we use that to inform what types of molecules we're going to make. But we're also academics, and we're George Jetson types, for those of you who remember the Jetson TV show, right? We are making things that we think will be valuable, even if there's not a market for it there. It's the if you build it, they will come, right? We are making, from lignin, products that the literature and the publication and the patent literature say are valuable precursors to build nylon. But there's no market for that compound because nobody has figured out how to make it. So I can't tell you whether a gram's going to be worth a penny or $1,000, right? So we're making those compounds and socializing that with industry to see what the real interest would be, either as is or if we can modify it microbially, or we can pass it off to a chemist to modify it to make those types of compounds. So there's a sweet spot there. We think of both of those things. Yes?
That's a wonderful talk. I'm really glad to see that the U.S. government is making such a long-term investment in a renewable source. And I'm sensitive to the fact that we have basically this constantly increasing carbon level in our atmosphere because we're releasing carbon that was sequestered many, many years ago. So biofuels would give us an opportunity to basically keep the same carbon budget in the air and recycle it year to year. But that only works if we have basically an energy in, energy out, unity or greater biofuel. So do you have sort of projections for the energy cost it takes to produce the same unit of energy from a biofuel, and is that going to be efficient once we scale?
That's part of our field to product. So the question, I think, boils down to, have we thought about this from a life cycle perspective in terms of what's the energetic and economic value of the things that we're getting out compared to how much needs to go in to grow the biomass and ship it and process it. And the answer is, we are thinking about those things. We have thought about it in the first 10 years in the context of making ethanol and not doing anything with the lignin to make chemicals. We are just beginning to do that analysis now on these types of integrated refineries. But I think some of the general principles from 10 years, so the jury is still out. Some of the general principles from the first 10 years will still apply. If we can use perennial crops that don't require fertilizer and pesticide input, the life cycle balance will ship in our favor, right? What the economics is will depend on what the molecules are that we're making. Is it only a gasoline replacement, or is it a gasoline diesel replacement? What's the selling price for fuel, and how much value can you make out of the chemicals? And how much energy does it take to do all of that? We know about the energetics of making ethanol, because as you all know, if you make ethanol, you put it in a distillation system and you distill it off. So I can run the numbers and calculate that. Many of the molecules that we're making will not be volatile enough to be distillates. We'll have to figure out other separation technologies. So until we make them and know what else is there, we can't really model the economics and the energy inputs of what those are going to look like. But we're thinking about all of those things with our friends in chemical engineering departments that think of process modeling.
Maybe the last question for Rob.
Yeah.
Two more. Okay.
Sure.
Professor Donahue, I have a question about modifying the microbes to tolerate inhibitors, the toxins. How far do you think we are away from being able to genetically engineer the microbes to tolerate all the toxins? Or do you think eventually we'll still likely need a dedicated separation process to remove the toxins?
I think we are very close to saying we have yeast and bacterial strains that can be resistant to the toxins we're going to see using the kinds of pretreatments that we're generating. As I said, the Zymomonas bacterium, which happens to live on plant tissue, seems to be inherently resistant to many of the things that choke Saccharomyces. Industry likes Saccharomyces as a biofuel producer. But we've been able to reverse engineer around a lot of that, at least in ethanol production. We are just beginning to make other molecules. And I think it's important that we remember that making a lot of any of these molecules is a toxic situation for microbes. They have not evolved to do it in their native ecology over millions of years, right? So making isobutanol or making a terpene at industrial level is going to put stress on the microbe. That's a compound stress on top of what they're getting from what they're seeing from the biomass. I may have figured out how to deal with the stress from the biomass when I make ethanol, but in a combinatorial way, that solution may not work when I make isobutanol or terpenes. The jury's still out on that one. That's why we need more money from the taxpayers to do basic research. I mean, those are the challenges. We don't know.
So your list of crops from the prairie to the cornfield leaves out wood.
No, there was poplar in there.
Well, okay, so is poplar special, or are all the...
Poplar is special because it's a rapid, it's something we can go in and genetically modify by transgenics, and it has a rapid recycling time, and it leaves roots under the ground and it puts off shoots. It's not like pine, which has a 30-year time before it gets big enough for someone to harvest. I can harvest poplar in 3 years.
But would all this work on pine, for example?
We know the GVL pretreatment works on pine. We know it works on softwoods. We know it works on hardwoods. But if we're going to engineer them and think about real bred or transgenic trees that have a 30-year time before they're mature enough to be harvested, I'll be pushing daisies myself by then. So it's going to take a long time. And it's also true, I think, I'm gonna say one other thing that I haven't said before. We know we can modify poplar and make transgenics. And so far, we've gotten USDA approvals, and we do it responsibly, and we've heard no complaints about that. I don't know that society worldwide is going to be comfortable with us making transgenic pine to be used to produce energy. There will be some crops in this 1 billion tons that are as Mother Nature gave them to us. And then there will be other crops that we will go in there and engineer to do things. And what the mix, whether it's 50-50, 90-10, 10-90, I can't predict right now. So pine might be one that's a latecomer, and we might want to go really carefully if we think about making genetically modified pine or other types of woody biomass. But if I can process wild type pine the way it is now, it works. Yeah.
Okay, let's thank Dr. Donahue again.
Thank you very much.